Welcome to the light microscopy lecture series. We will be taking small sessions and gradually we will build the concept of light microscopy. So to start with, first session is on nature of light. Okay. So just to give you a brief historical introduction as how the microscopy became so important tool and how people have used this tool to empower uh, unknown explored world where people had enough curiosity, but they didn't had enough information to get an idea. So a small example, in the mid 19th century, there was an outbreak of tuberculosis, similar to the pandemic situation we are facing right now. And in a reaction or in a way to fight out with that tuberculosis outbreak, people and scientists from different parts of the world, they started working, investigating on the causative microorganism behind this. So similarly, people have spent a lot of hours, a lot of effort into understanding these causative microorganisms. But the important thing is they always wanted a device which would help them to see something which is otherwise not possible to see with our normal eyes. Right? People were dying, millions and millions people were dying. And then there was always a quest to find the treatment. In such scenario, if we see the things have taken a turn when an Eastern German physician and scientist, Robert Koch, came up with a hypothesis or with an idea and established that bacterial techniques to diagnose bacterial infections. So on the evening of March 24, 1882, Robert Koch presented his landmark lecture with the statement on tuberculosis. So to quote, if the importance of disease for mankind is measured by the number of fatalities it caused, then tuberculosis must be considered much more important than those feared infectious diseases and such as plague, cholera, and so forth. So in the end of the decade, Koch conclusively was successful to establish that a particular germ could cause a specific disease. So this hypothesis was successfully established by him, and he did this by experimentation with anthrax. So using a microscope, Koch experimented with the blood of cows, and then he said, look, I have found something which causes this anthrax. So he observed something called rod-shaped bacteria and suspected they caused anthrax, right? So he discovered the causative organisms of anthrax, and not only that, the causative organisms for septicemia, tuberculosis, and also cholera. The cholera causative organism, which according to Koch was tubercle bacillus. Now, this was something altogether people were provoked to think onto a new dimension, an unexplored area, but it was only possible with the use of a microscope. Another important or a milestone achievement, which you might say, is in the year 1981. You can see here, Santiago Cajal was a Spanish scientist, basically a neuroscientist, pathologist, and was precisely a histopathologist. So what this Spanish scientist did, his specialization was on neuroanatomy. So, and he was working on the central nervous system. He, along with Camillo Golgi, used plan apochromatic objectives for the first time and then showed this beautiful picture of the neurons. So first time we could see the picture of neurons with the cell bodies, the axons and the synapse, everything was very clear. And because of this achievement, in the year 1906, they received Nobel Prize in Medicine. Now, why I'm trying to tell you all such things? Okay, so the important aspect is, no, the microscope has taken a very, very important role, and from time onwards, especially from the beginning of the 19th century and so on and so forth. But to understand the basics of light microscopy, just have a look at these two basic types of microscopes. So according to these two basic types of microscopes, what we say, the first one is as you can see, is basically a monocular microscope, because you have just one eyepiece and you're using usually one of your eye to visualize your sample. Whereas the other, the black microscope, which you can see, black colored. So this particular microscope is basically a binocular microscope. So why binocular? Because it has two eyepieces, and by using these two eyepieces, you can use both of your eyes to visualize your sample. So that is why two, and the name is binocular. Now, when we are talking of light microscope, the main important aspect, or the main you know is light. So because as the word says, light microscopy. So here in this case, if we are talking of light, and if you're talking of light microscopy, then I think we should have a very good understanding that what is light, why light is important, and what is the behavior of a light. So to start with, light basically acts as a messenger, right? And it transports the object information, as you can see here, from the specimen through the microscope. Okay, so from the specimen to the microscope, you can see your sample or your image is being created. But this translation of the object information in microscope in the form of an image of the specimen is again is visualized. Okay, so you visualize that image formation through the help of a microscope, and we visualize the image of the specimen, and we never visualize the specimen in itself. So important is what information the light has to give to us. To understand that, and how we can capitalize the property of light to get more and more information about the sample. Now, only the best management of light allows the translation of the information as accurately as possible from object into an image, which we call it as the final observed image. So again, as we are not visualizing the specimen or the sample itself, we are visualizing an image which is created by the light, right? And this particular image is a matter of concern for us. So to see how we can capitalize different property of light, so we need to have a better understanding of light. To have an effort, or to understand basically the property of light, we see a small cartoon animation here, and in this cartoon you see this is a bulb, and this bulb is basically, you can see waves coming out, and it is getting propagated, as you can see, with the yellow light. So this is basically in wave form or in particle form. Now there has been a debate, long debate on this, whether light is a wave in nature or light is particle in nature. And we will see one by one, because based on the wave property of light and based on the particle nature of light, we have capitalized them based on these two properties, and how we have capitalized to the benefit of our image acquisition we'll talk in depth. So first thing is, in general, I think most of you must be knowing that electromagnetic waves are there, and electromagnetic radiation which moves or which travels in the speed of light through vacuum is what we call as light, right? And it is, as you can say, it has both wave as well as in particle in nature. Now, this concept of like the light being an wave in form or particle in form came into existence after a long time, and there had been a lot of debate on it, and each and everybody had tried their level best to prove their theory as one. So one of the earliest person or the pioneer, Sir Isaac Newton, he in the beginning of the 19th century, he came up with a theory that is called corpuscular theory, and then with that he said light was considered to be streams of particles. So basically he was emphasizing that light being a particle in nature. And we, because of that, the entire, the community, people have started thinking light being particle in nature. And he was the chief architect of the particle theory of nature. So he believed that particles left the object and stimulated the sense of light upon entering the eyes. But things changed, and very soon in the year 1801, Thomas Young, so he came up with a very clear demonstration that, see, light doesn't have a particle in nature, or what he explained is light, on the contrary to what Newton said, was light had a wave nature. So he demonstrated with his demonstration, he proved that light behaved as wave nature, right? So he basically used light rays and used them to interfere. So when light from different sources, they interfered with each other, we could see patterns, and the patterns are in the form of crest and trough, which you see in the waves, and that is why we saw that patterns. And then he said that such behavior is not possible for anybody to explain with the particle theory of nature. So we need to have, or the light is supposed to have wave nature, and that is why with the wave nature you can explain this theory. Now he confirmed, yes. So he provided evidence, and with this evidence, he also explained the theory. But as time went by, people became more and more firm with the theory, and then they started believing on the wave nature. So one of them was like Maxwell. So Maxwell was also sure that light has a form of high frequency electromagnetic waves, and later Hertz also confirmed Maxwell's predictions. So what Hertz actually did, basically Heinrich Hertz in the year 1887, so he again started questioning. So he said, what I understood or what he found through one of his experiment was, he said that he has discovered the shining light of a metal caused electrons to be ejected. Basically, what happened when light was fallen onto a metal surface, he could see some shining light. So that which he explained that when light, basically a form of energy in particle form, when it is hitting onto a surface, it is knocking off one of the electrons. And this theory again, or this particular experiment again, triggered the heated or much talked about discussion and questioning on the wave nature of light. So he again, he said, these electrons were ejected, and how they ejected? They depended upon the frequency and not the amplitude of the light. So they were ejected because based of the frequency of the incident light, it didn't depend on the amplitude of the light, right? So why I'm saying this? Because energy is, as if you see on the wave nature, or if you see in the particulate nature, whatever energy is there, it basically is corresponding to the amplitude of that. So when you talk of the amplitude, which I'll show it later when I'll show you with the diagram, you can see that based on the amplitude, the higher the amplitude, more is the energy associated with it. Now, Einstein, in later that year, so he also seconded the Hertz discovery, and he said, okay, I have explained, I would like to explain what Hertz discovered. So he said electromagnetic waves can behave as a particle, and each of these particles are known as photons, or which is termed as photons. Basically, photons are small packets of energy, and this photons dependent, so they are energy dependent on the frequency of the waves. So this particular photons, they are associated with the energy, and this energy is dependent on the frequency of the waves. That's what Einstein seconded. Now, if you see this particular two diagrams, the first one, if you see the first one, in the first one, if you see, this is particles, and based on the height, so this is basically the amplitude. So if higher the amplitude, more the energy is associated with it. And this is also in the particles. You can see small circles there in the particle format, but they are moving in a pattern of wave, right? So this is basically in the pattern of a sine wave, which you can see plus and minus amplitudes are there. And why we're talking of the term electromagnetic waves. So when you talk of electromagnetic waves, if we see the electromagnetic radiation, so this is basically E is the electrical part and B is the magnetical field. So red is basically, you can see the electrical part, and B is the magnetic field, which is in transverse. You can see that is in blue color. So red and blue, this two goes together, and they are together known as electromagnetic waves. So the dual nature of light is, yes, possible. And how it is possible to explain the dual nature of light? That is by the electromagnetic waves. So light behaves both in the form of waves and also in the forms of particle. And as I was showing, so you can see all the red up here, they are all the electrical fields, and the blue on the transverse side, they are magnetic fields. Now, if we talk of electromagnetic field, so basically we should know the range of electromagnetic spectrum. So spectrum is basically the entire range of electromagnetic waves. And if you can see here, this portion, so these are different wavelengths in the electromagnetic waves, and these different wavelengths in the electromagnetic waves, they start from gamma rays, X-rays, ultraviolet, coming to the visible spectrum, to the infrared light, and to the radio waves. So why this entire spectrum is so very important? Why this is important? Because of this type of radiations. Now, if all of you can see this, you see here, I'll talk of two things. First, see here the wave. So this is basically wave. This is the crest, and this is the trough. So these are like basically sand dunes. When you when the top of the sand dune is known as the crest, and the bottom of the sand dune is known as the trough. So when you see the distance between two crest, you see it is very far. But as we move, you see from the radio waves, as we move to the gamma waves, we see the distance between the crest decreases, right? And that is why the wavelength decreases. So if I talk of like going from like radio waves, so the radio waves, basically these are the waves in the frequency here. These radio waves are used for radio transmission. Like we have been using televisions, we have been using FM, AM. Basically, we have been using microwaves. So we are using this particular wave to heat, for cooking, for radar detections in telephones and signals. Then comes the infrared radiations. Infrared are basically used to heat. So infrared radiations are used as room heaters, and they generate heat in your room. And then the next wavelength or the bandwidth comes for the visible spectrum. So visible spectrum is the spectrum or is the band of electromagnetic spectrum which is visible to our human eye. So without any aid or without any gadget. So which is visible to our eye, that is called the visible light. Now, as we move ahead from the visible light, we see the ultraviolet light, that is the UV. Again, UV, this apart from the visible light, none of these wavelengths are visible through our unaided eye. So we have been, we know the UV. And again, if we move, then the X-rays, they find applications in medical imaging. So where we find bone fractures and everything, we can use X-rays. And then comes the gamma rays. So gamma rays basically used in medicine for treating cancer patients. So in the form of a gamma knife. Now, why this electromagnetic spectrum is important? Because first of all, we can exploit the different wavelength or different region of this electromagnetic wavelength, and we can get more and more useful instrumentation, or we can use it either in medicine, therapeutics, we can use it either in our day-to-day life in the form of television, radios, and everything. Right? So we can exploit, until unless we know this property, we would have never exploited them. Now, again, so once we have known this, what we have been talking is about the visible light. So our entire focus should be on the visible light. Now, in the visible light spectrum, which starts something around 400 nanometers, so it says 380 nanometer to 700 nanometer. So this particular bandwidth is the visible light spectrum, which is visible to the human unaided eye. So we can see, and I think if you can recall your school time of the light concept and everything. So if you recall that, then we must have all learned about VIBGYOR, right? So violet, indigo, blue, green, yellow, orange, and red. And if you see here, so from violet to red, that is the spectrum, that is the distribution of the visible light, starting from lower wavelength to higher wavelength, that is from 380 nanometer to 700 nanometer. So you are going to exploit this visible light for, and we're going to capitalize this visual spectrum of the light to get information in light microscopy. Now, as I said, so we start something around from 380 to 700. But what is more important, within the visible spectrum also, there is gradually, if you start from 380, the lower wavelength, to higher wavelength towards 700, we see there is a gradual increase in the wavelength. So the wavelength increases. Now, why it is important to understand the wavelength? Because as the wavelength increases, the energy associated with a particular wavelength also decreases. So wavelength is inversely proportional to the energy associated with it. Now, if I say a smaller wavelength will be associated with a higher energy along with it, then I am correct. And if as we move, as our wavelength increases, the associated energy along with the wavelength also decreases. So why it is important? Again, because when we will talk of fluorescence microscope, how and what is the percentage of light, or what is the percentage of this wavelengths we are going to use it. Because if the energy associated with this wavelengths are high, we are going to use it for a shorter duration. If the energy associated with this wavelength is low, then we have to use it for a longer duration to get that emission signal from the fluorophore. So we'll come to that in our other sessions as we move ahead in our lecture series. But today, if we are talking of the electromagnetic spectrum, we have to keep this in mind, that energy is inversely proportional to the wavelength. Shorter the wavelength, higher is the energy associated with it. Okay. Now, to summarize the entire electromagnetic spectrum, light waves or the electromagnetic waves, they are transverse, as I showed. They have both. They have the electric field, they have the magnetic field. We will be talking of the magnetic field sometime later, because magnetic field doesn't have association here in this case. What we will be more worried, or we should be concerned about in case of light microscopy, is the nature of the light. So basically the wave nature of the light or the particle nature of the light. As and when this dual property of light, how it behaves when it passes through different lenses, will gradually understand that. Now, the electromagnetic waves, they can travel through vacuum. So they can travel through vacuum, and in vacuum, they travel with the speed of the light. And they show properties like reflections, refractions. They also have, the wave number, you can count it, that is with this expression 2 pi by lambda, where lambda is the wavelength of the wave, and the frequency, which is f, the frequency of this wave is also found through this expression, which can be calculated with the use of the angular frequency. Okay, so briefly summarizing, electromagnetic waves, they comprises of magnetic as well as electrical fields. They are transverse in nature. They can travel through vacuum. They travel with the speed of light, right? And the most important part is light, the electromagnetic waves, they have dual nature. So they have both the wave nature, and they have also the particle nature. Okay, so these properties of light we are going to exploit, and as we go from short wavelength to higher wavelength, the energy associated with the wavelength decreases. Okay, and the wavelengths or the wave nature of it can be, we have to distance, the wavelength is counted from the crest of one wave to the crest of other wave, and the amplitude of the wave also is directly proportional to the energy associated with it, right? So with this, the final thing is, in the electromagnetic spectrum, we have the visible spectrum, visible light spectrum, which ranges from 380 nanometer to 700 nanometer. And this is the most important bandwidth of the electromagnetic spectrum, because based on this, or capitalizing this bandwidth of the visible light spectrum, we are going to collect information in light microscopy. For more such videos and lecture series, please stay tuned to this channel, like, share, and subscribe.